So good morning, good afternoon, and good evening, everybody. Welcome to the Sanford Burnham Provost Cancer Center's Focus On seminar series, connecting established investigators with rising stars and addressing important questions about cancer. Today's focus is on cancer genetics, and our presenters will take deep dives into how nature's experiments on humans can lead to an understanding of cancer evolution and the development and resistance to therapy. Each presentation will be followed by a Q&A session, and if you have questions that you'd like to have addressed by the presenters, please type them into the Q&A box or the chat box. And Swasti Haricharan, the seminar coordinator, will come and monitor those Q&A sessions. So with that, let me introduce our first speaker. Dr. Michael Hoffman is a senior scientist at Princess Margaret Cancer Center. He's also an associate professor in the Departments of Medical Biophysics and Computer Science at the University of Toronto. Dr. Hoffman's research creates predictive computational models to understand interactions between genome, epigenome, and phenotype in human cancers. Dr. Hoffman trained at the University of Texas in Austin, University of Cambridge, and the University of Washington in Seattle. He is a new investigator awardee of the Canadian Institutes of Health Research. Michael, the floor is yours.
Thank you, Guy, for the kind introduction, and thank you, Swasti, for the for the invitation. And I see some familiar faces in the attendee list, so thank you, folks, for coming by. Some SVP folks, also some people from Twitter. If you're on Twitter, you could see me there. Feel free to tweet whatever you want on this talk, since it is going to be recorded. Or you can put it on Instagram or whatever, just not TikTok, because like I'm too old for TikTok, so please don't put this on TikTok. So I'm going to talk about HPV today. First, I don't have any competing interests related to this talk. So let's talk first about the standard model of how HPV genes are regulated and how HPV results in oncogenesis. This is the model that you will find in most of the literature. I've shown you here an example of an HPV genome. This is the HPV 16 strain, which is exactly 7,904 base pairs long. Each HPV strain might be a little bit different, but we're going to use HPV 16 as a model for most of the stuff I tell you about in this talk. So basically, the standard model is that you have HPV. It comes into a cell. The HPV genome is usually circular, and there are little circular HPV chromosomes in the cell, and there are these early genes, the E genes and the late genes, and the early genes start being transcribed initially using host human transcription factor machinery, and that causes these various genes to be expressed, like E6, which causes degradation of p53, E7, which causes activation of the host E2F1 transcription factor, which results in a cascade of other undesirable effects occurring, right? And also E2, which then binds to this region of the genome at the end, along with E1, which causes the replication of the HPV genome, and L2 expression, which produces the capsid proteins that are needed for HPV to leave the cell and go on to other cells, right? So based on this model, one would expect to see early gene expression, and especially E6 and E7 to show up in a big way in active HPV, right? And so we've looked at 5. I can show you an example from 5 cell lines. Here are E6 and here are E7, and there's relatively high transcription. On the y-axis, we're showing an example. We're showing a value that quantifies gene expression, basically. All right, and what I'm going to be showing you today is some work by my former PhD student, Mehran, who is now graduated. This was part of his thesis, and Mehran was looking at HPV HPV patient data from the Cancer Genome Atlas, and you can see those genomes that he was looking at here. So about 300 cervical carcinoma samples and 58 head and neck carcinoma samples. And so in the majority of cases, you see just what you see in these cell lines over on the left, which is high E6 and E7 expression. But he also found a number of cases where E6 and E7 had low expression, even in these active HPV-driven cancer types, and started wondering why that was. And so, started looking at what are other factors that can contribute to tumorigenesis that is not E6 and E7 expression. And one thing that he noted is that HPV integration is common in tumors. So while this standard model is that you have all these episomal chromosomes of HPV, you also find the case where here's a human chromosome and you find a little bit of HPV gets copied into here, right? And this isn't believed to be the predominant way by which HPV promotes oncogenesis, but Mayron decided to start looking into, maybe there is some sort of mechanism by which that integration can promote pro-oncogenesis. Do you have to include the important disclaimer here that even though we have low E6 and E7 expression in our data, it's possible that there was higher E6, E7 expression earlier, and then the steady state expression was lower in these particular cases. But nonetheless, we'll look further into this hypothesis, and you may see what we saw is that there's something related to the integration. So here's Mayron, who did all of the work that I'm going to tell you about today with our collaborators from the Machi Lupien lab at Princess Margaret Cancer Center and the Scott Bratman lab in Toronto. And he took the 5 HPV-16 cell lines derived from cervical cancer patients, which we list here. He wrote a new software package called Bellerophon, which identifies HPV integration sites by looking for chimeric RNA-seq transcripts. So basically what I'll be looking for is, you do RNA-seq and you have a transcript, and part of the transcript maps to the human genome, and part of the transcripts matches to the HPV genome. If you find a lot of these sorts of transcripts find a lot of reads where you keep finding the same sort of division in these chimeric reads, you always find an integration site right here, well, then you can conclude that there is probably integration into the genome at that location. And Bellerophon, which is free software that Mayron wrote, will do that for you. And so we had RNA-seq data, and we got ATAC-seq data, and on these 5 cell lines, and ChIP-seq data for CTCF, the so-called master weaver of the genome, a really important transcription factor that controls how the 3D structure of the genome works. And I want to show you here an example of what things look like around a integration location that Mayron has identified. So we're looking at this one cell line, HMS001. Mayron identified that on chromosome 20, position around 47 million, there is a integration site. And here, so we have a little correspondence between the genomic scale, and here we have a scale that is 200 base pair regions overlapping ATAC-seq peaks. So this is just a way to show you all of the ATAC-seq peaks in this region, which are going to be a little bit nonlinear. There are going to be clusters of them in some place, and less other places, and that's why we have this little correspondence here to let you see more of the ATAC-seq peaks on top. So, what you can do the same sort of thing for CTCF ChIP-seq data as well. So this is the same site, right? And then you can see up here what the ChIP-seq data for this cell line, HMS001, is compared to the other cell lines where we don't have integration at that position. So one thing you might notice is that there are, forward too fast, there are places where you have a different pattern in this cell type where we have the integration than the other cell types. So at the very bottom, we have this comparison between HMS001 and the extreme value from some other cell type. Right, so this is showing that there's more CTCF binding here than the other cell types. This is showing that there's less CTCF binding here than the other cell types. Right, there are a lot of places where you don't really find any change, but there are places where you do find change. And while this is one example, I'll show you later on that this is kind of a general trend for a lot of these individual HPV integration sites, is that there are changes in the local epigenome around it. So for example, this is the same HPV integration site right here, right? And we are looking at we're looking at attack-seq data, which indicates whether chromatin is open, and you can see that there's a lot of signal of open chromatin in this region, but only in HMS001. So other cell types, which are also cervical cancer cell types that have HPV integrated, do not have this, right? And you see kind of a similar sort of thing for the CTCF. You see these interesting peaks around here only in this cell type, but not on the others. And you see a similar story for RNA-seq as well. So you see there's this transcribed region here right at the integration site, but only for the cell type for which there is HPV integration at that location rather than other locations. We wanted to see whether this was something that was significant. So we did a permutation analysis. So basically what we do is in these 5 different cell types, there are a number of different integration points. And we simply scramble which cell type we assign each integration point to, right? So here's let's go back to HMS001. We have this integration site on chromosome 20, right? We just scramble things around so that we treat the data as if the integration site were instead on CHA, but we leave all of the other data where it is. And then we see whether these sorts of, these increases we seem to see at these locations, whether they have something to do with that location in a HPV-16 positive cervical cancer cell line in general, or whether there's something specific to this one cell line with the integration site right there. Right. So we can do this comparison around the integration site, looking, say, at 1,000 base pairs around the integration site. What you see is that there is a bigger open chromatin signal in the cell type with the HPV breakpoint right there than in other cell types. There is signal for CTCF that's not in other cell types. There's much bigger expression there than in other cell types. Right, and you see the same sort of trend if you look at the 1,000 to 10,000 base pair region away from the integration site. You see it a little bit in the 10,000 to 100,000 base pairs away. But once you start getting further away, like more than 100 kb, you look at 300 kb, 500 kb, right? Things go very much back to normal. It becomes relatively indistinguishable between the cell type that we're looking at and all of the other cell types, whether you shuffle things or not. So basically the hypothesis we get out of here is that the transcriptome and the epigenome will change near an HPV integration site in these cell types, but only up to 100,000 base pairs away. So this is kind of a local effect. So you go further out, the chromosomes regain their traditional regulatory patterns. So what might be generating this? So one unique feature of HPV is that there is binding of CTCV close to an integration hotspot. So if you do a motif scan with FEMO, just looking for the CTCF motif and a lot of different HPV genomes, you will find that in most of them, there's a really good match to CTCF around this position in the genome. And if we look at our CTCF ChIP-seq data for these cell types as well, we find that there is a good signal that that motif is actually driving the presence of CTCF in all of these different cell types. All right. So our results suggest that this CTCF finding might alter the native genome organization of the host. If CTCF binds in a place where it wasn't before, that can disrupt any sort of local chromatin loops or any topologically associated domains, and have relatively far-reaching consequences in the 3D organization of the genome, right? So something that you might expect to happen in the 10 to 100 kb range, but not further on. So we wanted to look next at whether these changes are able to promote tumorigenesis. And one thing we wanted to look at is, sure, we can see these changes in epigenome and transcriptome at kind of a signal level, but does this affect any particular genes? Does this mean that genes, potentially oncogenes, actually have higher expression, or tumor suppressor genes actually have lower expression when this occurs? So here's the analysis we did in these five different cell types. We look at all of the different integration sites. So you might notice here that some of them, like 93VU147T, has about 8 different integration sites, while HMSO1 that I showed you at first only has 1 integration site. And we look at all of the genes within 500 kb in both direction, and we look at whether the genes have a twofold change in expression and a difference of at least twice the standard deviation from the other cell types. And these are the ones that we have as non-transparent squares here. And so there are a number of genes that in different cases, in each integration site, you'll get different genes, have different expression, and that may drive something here. So I'll show you an example of the RNA-seq data for the individual genes that I selected on the previous plot. For example, FOXY1 is within 60 kb of the HPV integration site in SCC 090 and has very high expression. The other 4 cell types, it has almost no expression. EYA2 is a little bit out, 137, about 100 kb away from the HMS01 binding site, has high expression here, right? Low expression, very low in most of the other cell types, low in another one, right? Here's KLF12, which we see kind of the reverse pattern. In other cell types, it's high, but in CHA, it's low, with kind of this one exception here, right? We see the same sort of pattern for a lot of different genes over and over, is you have this interesting change of expression just nearby the integration site, right? And it only occurs in the cell line for which you have that integration site. We wanted to do some more rigorous testing of whether the changes are due simply to sample heterogeneity. So we did the same sort of permutation, and we're just looking at whether the transcription level in the cell type we're looking at compared to the transcription level in the other cell types, how that looks either based on the original data or where you permute things around so that the signal data, the transcriptome and epigenome data is the same, but we pretend that the locations of the integration sites are different. So basically what that gets you is a threshold that we can use to look at how sensitive this is to our threshold for what an outlier gene is, right? So if we set a really high threshold, right, we say that the fold change has to be above 8, well, we don't see anything, right? And we say the fold change has to be above 0, we don't we don't see the change in the right direction. But if we say, we're looking for a fold change between 2 and 7 log 2 fold change, then we see a separation between the original real data and the permuted data. And our conclusion from most of this is that HPV modifies the local transcriptome, right? There's a wide range in which you can define something as an outlier gene where we still see these sorts of changes. We wanted to look, so all of this I've shown you so far is on cell lines. And remember, this is originally inspired by TCGA data. So we wanted to go back and look at the TCGA data. And we find the same sort of thing in HPV positive cancer patients for which we can identify an integration site with chimeric reads. So in head and neck cancer and in cervical carcinoma, we see the very same sort of thing, where in the real data you find many more outlier genes at each threshold versus the negative control permuted data. You see this again and again, indicating that this is a mechanism that might exist not just in our cell lines, but also in these hundreds of real TCGA cancer data types. The other question we wanted to ask after this is this something that is related to integration of a virus into the host genome at all, or is it something that is specifically related to HPV? So we decided to look at Epstein-Barr virus, and luckily there's a lot of data out there for Epstein-Barr virus transformed cell types, for which we can get CTCF ChIP-seq data, and we can get DNA-seq data on where open chromatin is, right? And basically we see here, it doesn't matter which threshold you look at, there's not really a noticeable effect or certainly a significant effect in any of these cases. Right, so despite having integration of another virus, and we can kind of summarize this here. This is just a plot that shows that as you look closer to a HPV integration site, you find lots of significant changes in transcription, in open chromatin, and where CTCF is. Right, and basically you don't see those sorts of changes anywhere related to Epstein-Barr virus, right? So it's a much bigger effect in HPV if it's an effect in EBV at all. Finally, one other thing I want to show you is, if you remember that scan of where the CTCF motif is, we focused on HPV 16, but HPV 73 in that scan, you could see that it didn't have the CTCF sequence motif. And in these cases, in the HPV 16 case, we do not see the same sort of outlier gene expression pattern that we see in HPV 16 integration or anything else we've looked at where we have a good CTCF binding site inside the integrated HPV genome. So, we think that CTCF can affect the topological structure of chromatin here, and it definitely can in general. We think that might be what's happening here. CTCF can also directly affect alternative splicing. I'm not going to go into that here, but if you look in our preprint, you can see we've done a similar sort of analysis on alternative splicing, showing similar sorts of changes around where you have integration of HPV genome into the host. So I just want to finish up now. To summarize, we found that CTCF binds a conserved sequence motif in the human papillomavirus that modifies the local epigenome, possibly reorganizing topological domains. If you want to read more about this, you can find this on bioRxiv. You could just search for my name and HPV on bioRxiv if you can't remember this long string of numbers. But it is all up there, and if you have thoughts on the preprint, I hope you will send them to us. So I want to thank the people in the Hoffman lab. I especially want to thank Miran, who is not in the picture because he has graduated and has moved on to a postdoc working with people at Toronto's Vector Institute and with the Godarzi lab at UC San Francisco. Want to thank my collaborators at the Princess Margaret Cancer Center, and want to thank all of our funders. And finally, I'd like to thank all of you for your kind attention. Thank you very much.
Thanks very much, Michael. Before I hand over to Svasti to moderate the questions, I have a question for you. So my question is it possible to use this analysis to actually map the structure, the 3D structure of the genome?
Yeah, it's not really possible to do it like this. I mean, we could do there are experiments one could do on these cell lines for sure. That isn't what we did. I have a dry lab, so I have a limited capacity to do these sorts of things myself. And I'll be real. We were kind of hoping that we would submit the paper to a journal, and they would say, this is great. All you need to do is have the experiments that show that this takes place, because we really do think it is taking place. But they are, say high-C experiments are a little bit expensive to do. But no, it's consistent with every piece of data that we can find. And yeah, that's it.
I think Charles has a question too.
Michael, that was fascinating. Thank you very much. Really enjoyed your talk. Have you shown that any of these genes that are upregulated proximal to integration sites at the transcript level are functionally implicated in HPV tumorigenesis? Can you silence them by RNAi and show that you can just crudely affect proliferation or viral proliferation or what have you?
Yeah, let me.
Sorry.
No, I didn't say any of that stuff. It's not in the slides. I don't even have a bonus slide on this. But it is in the preprint. We did an analysis of essentiality for tumorigenesis and cancer survival for these different genes, the genes that are outliers here. So there is this resource that is called, oh man, I can't remember what it is.
Achilles.
Yes, Achilles. Yeah. That's it. Yeah. So we used Achilles. We looked at Achilles essentiality scores, and showed that there is a enrichment for tumor essential genes and the genes that are outliers here with a series score. I can share this briefly.
No, that's really exciting. It makes a lot of sense.
But along those lines, Michael, what about in the TCGA data? Is there anything different in survival outcomes or anything for the patients whom you think have this integration phenotype as opposed to the more canonical?
That is a good question. I don't think we actually looked at that. We did not look at that directly. We only kind of looked at it through the lens of Achilles and series scores. So that's a really good idea.
Yeah, also because I was thinking, you know what, I'll stop asking questions and I'll get to the audience questions, and I can always bug you about this later. So one set of questions that I think is really interesting is, what is going on with these specific genes that is it that they're more predisposed to have integration events? Or is it just a completely random process and it just so happens that sometimes it's in?
Yeah, that's our guess, is that the integration can happen anywhere. The integration is probably happening all over the place, but the only integrations we're actually going to see in a cancer sample or a patient sample are going to be those ones where the integration hit the jackpot somewhere where it's going to cause a oncogenesis that wouldn't have occurred otherwise. Does that make sense?
Yeah. So do you think there's any, and Charles will probably talk more about this soon, but do you think there's any interplay with DNA damage repair pathways in terms of how this integration is modulated, or has no one really looked at it and we don't know?
That is That is a very interesting question, which is outside my expertise. So either Charles will have to answer it, or I will have to take it back to our DNA damage people in Toronto to ask them, because again, it's very intriguing.
Yeah, I don't know, Charles, if you have an input on that. I don't know if we know that for any viral integration event, if there's any.
I don't know, I'm afraid, no.
Okay, cool. I found a new subfield now for us to work in. Another question, which I think is also cool, sort of evolutionarily speaking, what is the benefit to the virus of this CTCF binding and transcriptional upregulation? Is it some way in which it increases its own copy number, or is that also not really known?
Yeah, that's a good question. You know, I can only really speculate, which I will do so. And my speculation would be that the advantage is through this essentially what we think of as an alternative process for oncogenesis and reproduction. Like, I'm not sure that it does have a advantage in the normal case, sorry, the primary mode by which you have circular chromosomes in the host rather than integration. But given that the integration happens sometimes, you know, I think that we have shown a potential mechanism by which, in case of integration, there's a secondary mode by which the virus can do its thing and reproduce and go on. You know, viruses are very efficient in many ways, and sometimes have, you know, multiple ways of accomplishing something. So that's all I can speculate there.
Yeah,
if anyone else knows more, let me know.
Yeah, this is actually Peter Adams from Burnham is asking this question, and his point is really intriguing, because this virus is not evolved to cause cancer, right? So it's not like this is some sort of it just seems like a really weird synergy that you'd have these integration sites, and then you also have this increased transcriptional or genome modeling, I guess, there. But, you know, fair enough, we don't really know the answer. So what about exclusivity with P53 status? Do we see that the patients with the integration events are more likely to be P53 wild type or mutant? That
is another good question, and I don't believe we looked into that. So, you know, given what I was telling you about, you know, the role of E6 and E7 at the beginning, that is a good question, something that perhaps we should look into.
Yeah, also, yeah, I think it's.
I hope I can get a copy of these questions later so I can remember it. I don't have the wherewithal to take notes right now.
Okay, and last question, and then I'll let Charles do his set, and then we'll come back for a panel discussion at the end. So is there a known increase, because you showed this connection between the HPV insertion sites and head and neck cancer. So is that a known increase in these cancers after HPV infection? I think it is well known that HPV tends to be associated with head and neck cancer, right?
Yeah, there are a lot of HPV associated head and neck cancers. I'm not sure if the question is more specific about the, you know, host integrated HPV, and I'm not really aware that anyone has you know, shown something specific there with relation to head and neck cancer, but it is known as a you know, big cause of head and neck.
And it did seem like, at least in your analyses, even in the cervical cancers, you were seeing a similar subset of integration events, right? So presumably it is a common theme across the HPV associated cancers, I'm assuming.
Let me make clear that there are a lot of cases where it does not happen. So there are a lot of episomal HPV that is causing head and neck squamous cancer, and also cervical cancer. It absolutely happens without integration.
I want to see what you think about this huge subclonal diversity you're seeing, right? Where we're seeing some mutations in some genes that are subclonal. Do you think that they're less immune activating because you have these different types of cells, cheek by jowl, they tend to communicate with each other more, creating a secretome in the tumor that might potentially be anti-inflammatory or be turning the immune system off?
Yes, almost certainly, and that sort of ongoing work we're doing at the moment to try to establish what's happening in terms of cell-cell communications between tumor and normal cells, tumor and immune cells, to address that question. I mean, there's certainly a hint we're getting, and you know, it's early data, it's unpublished, but there is selection for sort of, I guess, yeah, an immune evasive phenotype in metastases, including the reasons I've shown you today, the HLA-LOH antigen presentation machinery disruption and neoantigen loss as well. As tumors evolve, they lose neoantigens and they select for sort of a more immune evasive genotype.
Yeah, and someone has actually asked a question that I wanted to ask you, which was, do you see any correlation between these clonal and subclonal mutations in mismatch repair defects?
So MMR defects is rare. MMR dysfunction is rare in lung cancer. I think in the 421 patients we've yet to publish, we've probably got three or four patients where we see a subclonal mismatch repair signal. That's four patient regions out of about 1,500 sequence. So it's quite unusual, but it is seen. But I guess arguably there are so many other ways with Apobec aging and the mitotic clock and tobacco carcinogens. It's not, you know, without wanting to put too anthropomorphic a view on this, it's not as if lung cancers are sort of short of a mutational signature or two. So that may explain why there's sort of no selection pressure for further ongoing mutagenic processes, perhaps.
And so why do you think there's this selectivity for some genes to be truncally mutated and some to be subclonal?
Yeah,
do you think
yeah, that's a great question. I think, you know, I think it's not just lung cancer. That's a question you could put to all tumor types. I think a lot of this comes down to cell of origin ultimately. What are the permissive cell of origin? So, for instance, in lung cancer, the type 2 pneumocytes, for reasons we don't fully understand, may be particularly sensitive to EGFR pathway induction, and that's why they're enriched for EGFR KRAS mutations. We don't know is the honest truth. Similarly, squamous carcinoma, more commonly we see copy number events clonally than we do mutational clonal events. Why that is, we don't know, and that may reflect underlying genome instability processes in the cell of origin, stem cell of origin, you know, tumor genome environment interactions we don't fully understand. I think we've got a long way to go to address those critical questions, actually.
And what do you think is, I know lung cancer is a bit peculiar because of the smoking as a huge mutagen. So do you think a lot of these things that you're finding are actually specific to lung cancer because of the smoking, non-smoking?
I think a lot of what we're seeing is sort of a microcosm of what you might expect to see in a late stage cancer, actually. That would be my guess. What you're seeing here is a lot of mutations in early stage tumors. But, you know, talking to my colleagues, Fabrice Andre, who's working on breast cancer and has got a large metastatic study, they see a very profound Apobec enrichment in the metastases and mutational burdens that aren't dissimilar to what we're seeing in primary tumors. So I think, you know, lung cancer is a microcosm for, in many ways, is an excellent tumor type to study because you've got copy number and point mutational diversity. Whereas in some tumors, many tumor types, you have one or the other. In lung cancer, you've got both. And so, you know, it's harder to study as a result, but I think you get to sort of the heart of the problem probably a little bit faster by studying lung.
And what is your take on why you would see the Apobec expression peak sort of pre-neoplastically and then go away?
Well, yeah, I mean, that's a jolly good question too. So, well, I didn't have time to show you the data in the bioarchives paper. So, first of all, we show that if you induce Apobec at the same time as you induce EGFR in that mouse model, that interestingly, it prevents tumor formation. So it's almost as if you have to get to a certain population size for Apobec to have an advantage, and in this case, in the context of EGFR therapy. But I think probably what's happening is that Apobec is driving an immune response that probably eradicates ongoing mutagenic clones too, which is why you dampen Apobec expression as tumors evolve. That's probably what I think is happening. We're interested, I mean, we're intrigued by the fact that the Apobec signal comes on at the senescence boundary. We show that in vitro models as well. It's a parallel pathway, probably driven by DNA replication stress. And we're wondering whether it fuels the ability of cells to circumvent senescence by generating chromosome copy number events that I mentioned through chromosome instability. So that's something we're working on.
Gosh, that's really interesting. I'm going to get to a few more questions from the audience if you have the time.
Sure.
And maybe Michael, I can get you to come on as well to give your input. One of the questions is this dichotomy between tumor mutation burden and epigenetic regulators, and how much do you think your cold tumors needing to be converted would actually need epigenetic modifiers?
Yeah. So I didn't have time to show today. We've done a little bit of work, not as much as Michael's elegant work on the epigenetic side of the spectrum. And what we find is that epigenetic silencing of neoantigens is also a mechanism of switching them off. We've only subjects about 50 or 60 tumors to RBS analysis, so it's early days, but we certainly see quite a strong signal of neoantigen repression through promoter methylation in tumors that lack any other mechanism of immune evasion.
Yeah, so
it's quite an interesting point, because you know that the reversibility of that may also explain why, if you retreat a patient with a checkpoint inhibitor, some patients do re-respond to therapy. So it may be in these essential genes, you're repressing them. The tumor, there's a detrimental impact, but in the presence of CPI therapy, it's advantageous. But when you stop CPI therapy, it's disadvantageous. So there's sort of a push me, pull you evolutionary tug of war going on there, perhaps.
Yeah, and in context of Michael's work as well, I know that there's been thought that it's not just the tumor mutation burden, but it's actually the types of neoantigens. So those that are more bacterial or more viral, or in other words, more other.
So I think, yeah, that's a good question. I'm a bit skeptical whether microbial neoantigens will be fruitful, because unless they're driving tumorigenesis. So I think for any of these neoantigens to be useful for the body, for the patient, they have to be, I think, required for tumor growth. That's a bit of a sweeping statement, but I think if we go down that route, we probably won't go far wrong, for the simple reason there are so many ways a tumor can rewire its genome, or the immune system can eradicate tumor cells with microbes within them. If those microbes aren't required for ongoing tumor growth, then the tumor will lose nothing by just eradicating a subclone of cells with the microbes in them. And similarly, with non-essential genes that aren't required for tumor evolution, if they are neoantigens, and we actually showed that as well, I didn't have time to show you, that there's heavy selection against mutations in genes as tumors evolve, in essential genes as tumors evolve, because tumors cannot afford to derive neoantigens in genes that are essential for survival.
Yeah, actually that also makes a lot of sense. Another question is that in glioma, we don't actually see an association between TMB and response to PD-1. And actually that's true in ER-positive breast cancer as well. So do you think that might be because we are not distinguishing between clonal and subclonal mutations? And that's really where we need to be focusing rather than
I think it's a jolly good question. And that could be one potential reason. I don't think it's the only reason though. I mean, for example, one thing that's always intrigued me is that GBMs that are treated with temozolomide develop MSH6 mutations, and they develop mismatch repair deficiency and an overwhelming tumor mutational burden at relapse. And judging from the my central nervous system medical oncologists I've spoken to, they rarely respond to CPI therapy despite having a huge TMB. That would be predicted to be subclonal. Is that the reason they don't respond? I don't know. I think there are probably more subtle reasons, like ongoing immune suppression and regulatory mechanisms we just don't understand yet. And I don't think, you know, when Kevin Litchfield in the lab modeled to what extent the genomics predicts all the variants accounting for CPI response, we're not even 3/4 of the way there yet. We're probably only just slightly over halfway there. So there is still a lot, probably tumor microenvironmental features, perhaps germline features, that are also probably important in guiding response. I think the other thing about GBM is you also have to remember that there's a lot of extra chromosomal DNA, and these extra chromosomal fragments can sort of come and go with cytotoxic targeted therapy and probably checkpoint inhibitor pressures. These ecDNA fragments wax and wane, and that's a huge challenge, I think, to therapy.
And what about, Michael, in terms of the viral integration sites? What do we know about how the immune system interacts with it? Does it at all, or?
That's an interesting question. You know, I don't have an answer with respect to the specific stuff I presented. it is interesting that there's been a lot of work in the last, 5 to 10 years on immune response to endogenous retroviruses and this so-called viral mimicry reaction, where a lot of what people think certain cancers are causing a innate response to something that appears to be viral within the cell, which I think is very interesting. But I don't know of anything more specific to my work.
Yeah. So one of the things
I'm afraid I need to go because I'm 10 minutes late for my next meeting. I hope you'll forgive me. But thank you so much. Thank you so much. Thank you so much for inviting me.
Of course.
Thank you, Guy. Thank you, Michael.
Yeah, I actually think that is a really interesting question, because I haven't read a lot in the recent literature about viral integration and how that would affect the immune system. It seems to be a lot more focused on mutations and so on and so forth, rather than what looks like epigenetic or genetic modification at the locus.
Yeah, I mean, there's a lot of, obviously there are a lot of genetic changes, and there are a lot of recurring genetic changes, and that's what you see all over TCGA, are the same genetic changes over and over again. But there are definitely cases for epigenomic pathways for oncogenesis. The other thing I like to think about are ependymomas, which are a kind of brain cancer where a Canadian group has shown that there is very little recurring genetic change in a class of ependymomas, but frequently similar sorts of epigenetic changes, which is a very interesting case. And certainly there are a lot of types of cancer, for example, IDH and leukemia, acute leukemia and glioblastoma, where epigenetic pathways are implicated, such as IDH affecting methylation pathways and things like that. Although that's a little bit different from what you were saying, because it's still a genetic change. But to me, the fact that that genetic change implicating a epigenetic pathway exists means that it's quite possible there could be similar epigenetic only pathways for the same sort of thing to happen.
Yeah. Well, I think if there are no more questions, Guy has no more questions, then we can thank everyone for taking the time to attend this, and thank you, Michael, for the great talk, the Q&A, the panel discussion, and for sticking around to talk to our trainees. I know they're really excited about that. And I would like to also remind everyone that the next Cancer Center seminar is on July 12th. And we'll have Dr. Campisi and Dr. Ed talking about senescence in cancer. And I think it's going to be a really interesting session as well. Thank you for joining us.
Thank you so much.
Thanks very much, Michael.